Wait till it's over, guys. See if you like it. So most of us don't spend too much time thinking about our livers. Unlike our pumping hearts and our reasoning brains, the liver is normally a silent, though essential partner in our lives. Right now, it is quietly working on our lunch, keeping us in metabolic balance. But when damaged by alcohol, infiltrated by metastatic cancer, or felled by a viral infection, it can no longer be ignored. Hepatitis C virus, or HCV, is one of the things that can bring the liver to our attention. The virus is now growing persistently in the livers of an estimated 170 million people around the world and over 3 million in the US. With time, HCV can destroy the liver's essential functions, since an infection is often a prelude to cirrhosis or to cancer. Today, we celebrate 3 people whose work has enabled medicine to eliminate infection with HCV. To eliminate, not just control, a chronic viral infection is unprecedented. And the story is unusual in other ways as well. First, why that C? In medical school, I learned about hepatitis A, a self-limited viral infection that was usually transmitted by food, and about hepatitis B, a chronic infection usually contracted at birth or by transfusion or intravenous drug use, often causing cirrhosis or cancer. Over the next 20 years, vaccines were developed to prevent these infections, although there are still many millions of people around the world chronically infected with hepatitis B virus. We developed tests for hepatitis B virus that makes transfusion safer. But a big part of the hepatitis story was clearly still missing. Harvey Alter at the NIH Clinical Center showed that another prevalent hepatitis virus, neither A nor B, must be in our blood supply. For example, he could transmit it to non-human primates and cause disease. But the usual methods did not help him to identify the so-called non-A, non-B hepatitis virus. Where traditional virology failed, molecular biology rescued us. In 1989, Michael Houghton and his colleagues at Chiron Corporation isolated a piece of nucleic acid from the blood of an infected patient, a patient infected with non-A, non-B virus. They found that it was part of the genome of a novel virus now called HCV that belongs to a known class of human pathogens. The virus was enough to design a diagnostic test for HCV, protecting the millions who receive transfusions each year. And in 2000, Alter and Houghton received a Lasker Clinical Prize for their work. That fragment of the hepatitis C virus genome could have been the toehold that led quickly to the production of abundant virus and to the design of a vaccine and antiviral drugs. But that didn't happen. It didn't happen because HCV is an odd and perplexing virus. And here is where today's heroes enter the story. Charles Rice had been trained at Caltech, working on the class of viruses, the flaviviruses, to which HCV belongs. So when that HCV fragment had been announced, he saw its potential. By then, he had joined the faculty at Washington University in St. Louis, and his laboratory aimed to do what could be considered the obvious thing, to extend that fragment into a complete viral genome, a starting point for making infectious hepatitis C virus in cultured cells. One could argue it should have worked easily, but it didn't. When he and some others finally found the missing pieces, he learned that variations in HCV genomes impeded the production of virus in cultured liver cells. Finally, he was able to puzzle together a genome that did make virus, and the virus could infect a chimpanzee and cause hepatitis, and this was a major achievement. But that virus still didn't multiply in cultured cells, so essential studies were stymied. Ralph Burtenschlager was working in a parallel universe, trained in Heidelberg, working on the hepatitis B virus. After joining a pharmaceutical company in Germany, Ralph also saw the opportunities that were inherent in Chiron's piece of the HCV genome. And then he experienced many of the same frustrations that Charlie Rice had experienced. And he and Charlie commiserated about these at international meetings for nearly a decade. After returning to the university in Heidelberg, Ralph and one of his trainees, Volker Lohmann, conceived an interesting plan. They would try to multiply only a part of the viral genome in liver cells. Now, the strategy precluded the production of complete infectious virus, but it allowed them to select partial genomes that could multiply efficiently. In turn, those efficient partial genomes allowed drug companies to seek compounds that inhibit the replication of the partial genomes and other viral functions. Now, let's remember that looking for antiviral drugs was not, and still is not, a trivial exercise. Before the development of drugs that inhibit HIV, the human immunodeficiency virus, few antiviral drugs existed. Treatment of viral infections, like HCV infections, if possible at all, usually depended on toxic drugs, like interferons, that bolster host defenses. Work with HIV taught us how to interfere specifically with viral enzymes essential for viral growth. So ambitious chemists got to work on the partial genomes of HCV, and Michael Sophia was among those. In 2005, Mike had just left a large pharmaceutical firm to work at Pharmacet, a much smaller company which sought drugs against a number of viruses, including HCV. One type of compound looked especially promising. It inhibited the viral enzyme that copied the HCV RNA genome, but it didn't behave well in animals, rapidly decaying or failing to get into cells. With a series of chemical tricks, Mike and his team turned it into a drug that was concentrated and then activated in the liver. That drug, now called sofosbuvir, turns out to be highly effective against many different strains of hepatitis C virus, is impervious to drug resistance, and has become the common component of the several drug combinations approved by the FDA over the past 3 years for treatment of HCV infection. These combinations can eliminate HCV from as many as 99% of infected patients over several weeks of treatment with minimal toxicity. Some benefits to patients have been immediate: improved liver function and loss of infectiousness. Others are very likely: the sharply reduced risks of ultimately experiencing cirrhosis or liver cancer. Now, everyone here will recognize that these results are extraordinary, but thus far the number of HCV-infected people has declined only slightly in this country or worldwide, because most have not yet received the drug. One reason, among several, for this is their cost. The public outcry about the cost has not escaped the notice of those of us who sit on the Lasker jury. And we have also noted that routes to wider access are being explored and sometimes used. But our job today is to celebrate those who walked a winding scientific trail that led to the miraculous drugs and to the benefits of using those drugs. At the same time, let us resolve to find the means to provide them to all who can benefit. And let us hope that by making these awards today, we have explained why it is so important to do so. Thank you.